In this tutorial, we will understand cell, the unit of life, and all important basic terms and definition related to this. So, cytology is the branch of biology which comprises the study of cell structure and function. And cell is the structural and functional unit of all living beings. There are two types of cells, plant cells and animal cells. Let's understand plant cell now. In plant cell, cell wall is present. Nucleus usually lies near periphery due to vacuole. Centrosome is usually absent from higher plant cells except lower motile cells. Plastids are present except fungi. Mitochondria is generally spherical or oval in shape. Single large central vacuole is present. Number of mitochondria are from 200 to 2000. Cytoplasm during cell division usually divides by cell plate method. In animal cell, cell wall is absent. Nucleus present near the center. Usually, centrosome is present that helps in formation of spindle fibers. Plastids are absent. Mitochondria is generally tubular. Many vacuoles occur which are smaller in size. Number of mitochondria is approximately 1600 to 16,000 in liver cells, and cytoplasm divides by furrowing or cleavage method. Plant cells are capable of forming all the amino acids, coenzymes and vitamins. There is no contractile vacuole. Sodium chloride is toxic to plant cells. Plant cells are generally well over 100 micrometer long. Spindle formed during cell division is anestral. Lysosomes present in less number and chromosomes are larger in size, where animal cells cannot form all the amino acids, coenzymes and vitamins. Contractile vacuole may occur to pump excess water. Tissue fluid containing sodium chloride bathes the animal cells, generally much smaller than 100 micrometer. Spindle formed during cell division are amphiestral and lysosomes present in more number and chromosomes are very small in size. Let's understand difference between prokaryotic and eukaryotic cells. Prokaryotic cell is a single membrane system. Cell wall surrounds the plasma membrane. Cell wall composed of peptidoglycans. Strengthening material is Murier. Cell membrane bears respiratory enzymes and cytoplasm lacks cell organelles, mitochondria, Golgi body. Ribosomes are 70S type. There are no streaming movements of cytoplasm. Endocytosis or exocytosis do not occur. Eukaryotic cells are double membrane system. Cell wall surrounds the plasma membrane in some protists, most fungi and all plant cell. Animal cell lack it. It is composed of polysaccharide. Strengthening material is chitin in fungi and cellulose in other plants. It lacks respiratory enzymes. Cytoplasm contains various cell organelles. Ribosomes are 80S type. Cytoplasm shows streaming movements and endocytosis and exocytosis occur in animal cells. In eukaryote, mitotic spindle is not formed in cell division. The mRNA does not need processing. Nuclear material is not enclosed by nuclear envelope and lies directly in cytoplasm. It is called nucleoid. And in prokaryote, mitotic spindle is formed in cell division. The mRNA needs processing. It is enveloped by nuclear envelope. Nucleus is distinct from cytoplasm. In eukaryote, DNA is circular and not associated with histone proteins. Replication of DNA occurs continuously throughout cell cycle. These have small size 0.5 to 10 micrometer and have much less DNA. Sexual reproduction absent but parasexuality present. Plasmids and pili occur in many prokaryotes, example E. coli. Cell division mostly amitotic. Plasma invaginates and from finger-like process. Mesosome which take part in respiration. In prokaryote, nuclear DNA is linear and associated with histone proteins. Extranuclear DNA is circular and protein-free. Replication of DNA occurs during S phase of cell cycle only. These are relatively large, 10 to 15 micrometer, and have much more DNA. Sexual reproduction is present. There are no plasmids and pili in eukaryotic cells, example, Spirogyra and Cholera. Cell division is typically mitotic and mesosomes are absent. Let's understand some very important definitions. First is cell wall. Cell wall is the outermost rigid, protective, non-living and supportive layer found in all the plant cells, bacteria, cyanobacteria and some protists. Every living cell is externally covered by a thin transparent electron microscopic elastic regenerative and selective permeable membrane called plasma membrane. Protoplasm is a complex, granular, elastic, viscous and colorless substance. It is selectively or differentially permeable. It is considered as polyphasic colloidal system. The substance occurs around the nucleus and inside the plasma membrane containing various organelles and inclusion in called cytoplasm. Mitochondria are semi-autonomous having hollow sac-like structures present in all eukaryotes except mature RBCs of mammals and sieve tubes of pholem. These are absent in all prokaryotes like bacteria and cyanobacteria. Plastids are semi-autonomous organelles having DNA, RNA, ribosomes and double membrane envelope which store or synthesize various types of organic compounds as ATP and NADPH+ and H+. These are largest cell organelles in plant cells. Endoplasmic reticulum. It is well-developed electron microscopic network of interconnected cisterine, tubulars and vesicles present throughout the cytoplasm, especially in the endoplasm. And last is Golgi complex. Is made up of various membrane system, example cisterine, vesicles and vacuoles. So they were all the definition related to the topic called cell, the unit of life. For the detailed topic, please go through our website www.Syft.in. Till then, thank you very much for watching us.